Howdy, this is our Lab 2 Aseptic Techniques lab report, and our group is number 2, Diego Castillo, and myself, Rita Cantu Mahurin. For our introduction, we are covering the main objective of performing aseptic inoculation techniques in lab. The materials we utilized throughout this lab as far as cultures included the E. coli in the tryptic soy broth, also known as TSB, the SRS on blood agar plates, the mixed culture broth, and we also utilized other media such as the 2 TSB plates, the 2 TSA 3 milliliter plates, the 2 TSA 9 milliliter slant plates, and the 3 TSA plates. As far as our procedure, we first started by inoculating the TSB with a loop with E. coli and SRS respectively. We then utilized the TSA 3 milliliter using a loop with E. coli and SRS. And then we inoculated the TSA 9 milliliters with a needle with E. coli and SRS. And we had to ensure that we were stabbing into the butt of the tube and streaking the slants as we pulled the needles up. And then we inoculated the TSA plate with a loop with SRS, and we streaked for isolation. Coming from a solid media, we see one colony. Inoculating the TSA plates with a loop with E. coli was also performed, and the final step was utilizing a TSA plate with the loop with the mixed culture broth and streaking for isolation, and we were expecting 2 different colony types from this specific media.
Howdy. So for our results of the SRS and TSB, the growth appeared at the bottom of the vial and it floated once shaken. Just throughout the vial, you could see obvious signs of cloudiness and different um, growths that were happening there. It is a little hard to see on the photo, but there is a significant amount of bacterial growth anyways, and technique appears to have worked. It could be improved upon maybe with more bacteria or maybe with more time for it to actually grow. But it was done by Rita, and she did get growth, so it appears to be good. Um, and it does show a little bit amount of growth throughout it, with it not really staying in one singular spot. And this one was done by me. This is the E. coli in our TSB broth. And there was notable growths in this one. In the photos, you can see the cloudiness of the actual um, vial. On the left picture, you can see the floating debris that's inside of the vial representing the bacterial growth. And one of the main things that we actually noticed with the help of Lucas and our other TAs like Patty and Marie was that there was a ring at the top of the broth, which is, after further research, is known as it could be pellicel, which is a type of biofilm that will be made on top of the bacteria where the air and the liquid would meet. And a reason behind why E. coli would have this specific biofilm is because it's a facultative anaerobe, meaning that it can grow with or without oxygen. Just that if it did have oxygen, it would grow much quicker. And the concentration of that growth in that area is just held together by an extracellular matrix. And that's what makes that little ring around the top of the liquid. This is our SRS and TSA 3 ml. This was done by me. You can see pretty significant colonies growing. However, one of the problems in my technique that I did is I did not inoculate fully to the top of the slant. So you can see about 3/4 of the way through of the um, growths that it just stops. And that's an error on my part. But other than that, you can really see great colonies in this one. And it's just really prevalent, and you can see the different growths. Better technique would mean that you can see it more, obviously, and TSA is pretty great for growing any type of microbe because it's not really selective, and any type of microbe can really grow in there, almost any type. And this is our E. coli and our TSA 3 ml slant. And this slant was done by Rita, and it showed great single colonies throughout, even to the top of the cap, um, which is it was a little close towards the cap, but it didn't actually reach it. But you can see in the pictures that it did reach all the way to the top. Um, it does allow for longer storage with these TSA slants because they're actually closed, and it reduces the risk of cross-contamination with either different microbes in the air or different microbes around. Just the TSA slants will do a much better job of actually storing. So this is our SRS and TSA 9 ml. This one, the colony's a little like um, thicker, and you can't really see single colonies as much. It could be an error with the technique, and the streak was continuous to the top of the tube and did show significant growth. There was a stabbing technique used in this part. However, the stab wasn't deep enough. It was only about an inch, maybe less. And in the photo on the right, you can see there's a little line where the stabbing occurred, but it wasn't deep enough to show significant results. The growth was still present, but it wasn't as great as the prior slants. And this one was done by Rita. So this is our E. coli and our TSA 9 ml slant. Colonies seem to be single, but they are really small, and you can barely see them in the photos. But when looking at them in person, you could actually really see them pretty well. Growth is continuous to the top of the tube, and there is some small growth seen in the stabbing portion of the butt of the vial. On the left picture, you can see there's this slight line there towards the bottom. It's still not deep enough, but it is deeper than the other, and that allows us to see a little bit of growth there. And as we've learned in our other um, labs, that it's mainly for motility to see if the bacteria will move. And overall, it was a decent slant with single colonies being present. However, it is a little faint to see, and if only the stab was a little deeper. Oh, and this was done by me. Okay, so these are our plates. I'll start from the top. SRS on TSA. So this plate was done by me, but it did not come out good. Um, First of all, there's significant growth on the first portion of the plate, and that is because either I grabbed too much bacteria from our sample and it just caused too much bacteria, or I inoculated too much. And then in the second inoculation, you can see there is more single colonies that are visible, but it is still too much bacteria. And then eventually it just disappears, and that is not right. It could be an error on the cleansing of the loop between um, cultures because I could have possibly not let it cool down enough, possibly killing the bacteria and spreading it. I could have just missed the bacteria in general, but overall it disappears at one point, and that is not good. So it was not a great plate and didn't show much that we needed to see. As for our E. coli, it was a little better. The initial inoculation does show those single colonies, and the only problem is that it is still too little, and there isn't enough inoculation present, and you can't really see as many single colonies, and reducing the amount. That was done by Rita. It did. She did a good job at the beginning, but then it just didn't do as well towards the end. And then finally, for the last one, our mixed culture on TSA. This one was one of the better ones at the beginning, and it does show single colonies and also the spreading to the next area and the next area. But it's still too little. Like there should be more colonies growing, so there were still errors on the technique. It could just be grabbing not enough layers or not inoculating close enough. But it did show the most single colonies from the two priors, so that one was one of our better ones, and that was done by me. So our results overview. These are some questions that we had that we wanted to answer. So why is it important to sterilize a loop between streaking the same plate with the same bacteria? This is to avoid too much bacterial growth because you do want to reduce the amount of bacteria growing throughout so that you can see more single colonies and see isolation. And that's the whole point of this is to see the isolation of those single colonies to identify what they are. And how did the inoculation technique differ between the broth, the slant, the deep, and the plate media? Well, for the broth, you kind of just grab the sample bacteria, put it inside the broth, and just swirled it a little bit to let the bacteria sit and grow. And that's the difference between that. For the slants, it was with the loop still, but you did loop sample from the bottom of the slant all the way to the top, and so that one was a little more of a just picking up your hand and moving side to side. For the deep one, the deep slant, we had to use a needle for that one so that we can stab the bottom of the vial. And that is to show the motility of the bacteria or the microbe. And so instead of using the loop like we did for the rest of these, we use the needle to grab our bacteria, stab the butt, and then proceed to pick up the needle and swipe it left and right up as we went up the slant. And then finally, for the plate media, well, that one required four different swabs or inoculations. And those were at the beginning, you would do about a third of the plate, rotate the plate, then grab two lines of the previous bacteria after sterilizing the loop, waiting for it to cool. Then you would grab the two lines of that media, move it across, then rotate once more, sterilize, move it across, rotate last time, and then sterilize and then move it across. So that was mainly to have isolation and lower amounts of colonies as you progress through the plate. The mixed culture plate differed from the pure culture plates because colony morphology was different. For ours, it was a little harder to see because of the errors that we made, but in theory, we should have seen differences in the single colonies. So some colonies would have looked this way, and the others looked a certain way. But unfortunately for us, we didn't really see that well. And streaking for isolation is very important when working with mixed cultures because you want to see the differences. You want to see which bacteria is which. And if you don't get isolation, you can't tell which is which unless you do other testing. And so isolation is very necessary to indicate differences in your microbes when you're working with it. And successful isolation on the TSA plate indicates great technique. It indicates that you did the correct steps and that you did it in with enough bacteria that it wasn't too much, but it also wasn't too little. And errors would affect the results as you saw in ours. It's either it's a too much of a heavy hand where too much bacteria was used, so you don't see any single colonies, or in the way that no differentiation is between them. So there's isolation wouldn't show and you wouldn't see a difference between them. And future improvements could just be just learning to how to do it better and accurately knowing how much of our bacteria samples we should use and keeping everything sterile. I think we did a good job on the sterile part, except for the part when I possibly sterilized and didn't let it cool down enough. But other than that, I think we kept everything clean and I think we did a good job of making sure that there was no cross contamination. But for sure, accuracy was a little bit off with our inoculating and could be improved upon.
Overall, in conclusion, in this lab, aseptic inoculation techniques were applied with E. coli and SRS using broths, slants, deeps, and plates, and this experiment highlighted the importance of using the correct tools, the loops versus the needles, sterilizing between streaks, and maintaining aseptic conditions to prevent contamination, and also assuring that there are accurate results. The differences in the growth patterns and colony morphology between the species as well as the mixed culture plate emphasized the value of streaking for isolation. The growth patterns differed between the E. coli and SRS, with notable differences in colony morphology and turbidity in the broth cultures. This experiment also used multimedia, the TSB broth for liquid growth, the TSA slants for surface inoculation and maintenance, and the TSA deeps for oxygen and motility testing. In addition, the TSA plates for streaking and colony isolation. Overall, the successful growth and isolation demonstrated proper technique and its importance, as well as the role of media type in studying microorganisms. From this lab, the aseptic techniques are essential in microbiology because they ensure that reliable results are always collected, it prevents cross-contamination, and it allows observations of microbial characteristics that are accurate. Thank you so much for listening. This is Group 2. And we appreciate you taking the time to listen to our lab report.